Traditional introductory biology often presents the eukaryotic cell as a microscopic soup, a water balloon where organelles float in an amorphous, passive fluid. This model fails to account for how a cell executes millions of conflicting chemical reactions simultaneously, synthesizing proteins mere nanometers away from enzymes designed to destroy them. Physical compartmentalization provides the structural solution to this paradox. The cell utilizes distinct lipid boundaries to create isolated environments that operate in parallel. These boundaries define the endomembrane system, functioning as a continuous, physically linked biochemical processing line. To understand how a cell actually operates, we must examine it across three distinct domains: its manufacturing production lines, its physical infrastructure, and its precise temporal gating. Organelle function only becomes clear when viewed through its connection to this larger logistical network. The production line begins inside the nucleus. This central information repository is physically isolated from the rest of the cell by a double-layered nuclear envelope. The nucleus organizes DNA into two distinct spatial regions based on activity. Transcriptionally active genes are kept loose as euchromatin, while silent, inactive genes are packed against the inner membrane as heterochromatin. When a specific gene is transcribed, the resulting messenger RNA sequence exits the nucleus through highly selective nuclear pores to enter the cytosol. This mRNA binds to a ribosome attached to the rough endoplasmic reticulum, or RER. As the ribosome translates the genetic code, it threads the raw polypeptide chain directly through the membrane into the isolated lumen. Initial protein folding and N-type glycosylation, the addition of sugar residues to nitrogen atoms, occurs strictly within this protected RER lumen. The adjacent endoplasmic reticulum lacks surface ribosomes and plays no role in protein synthesis. The smooth ER focuses on manufacturing essential lipids, like phospholipids and cholesterol, and utilizes cytochrome P450 enzymes to detoxify harmful xenobiotic compounds. Newly manufactured proteins and lipids are packaged into transport vesicles that bud off the ER and travel to the cis face of the Golgi apparatus. The Golgi acts as the central sorting and modification hub. As proteins move through its stacked layers, they undergo further complex modifications, such as O-linked glycosylation. Finally, at the transface, the Golgi dispatches vesicles toward their specific cellular destinations. Through this sequential assembly line, the endomembrane system transforms one-dimensional genetic code into fully functional, spatially targeted molecular machinery. The high density of the cytosol makes passive diffusion an inefficient mechanism. To move material, the cell relies on an active physical infrastructure, the cytoskeleton. This highway system is built from rigid tubes called microtubules, constructed from alternating alpha and beta tubulin proteins. Specialized motor proteins navigate these tracks by burning ATP. Kinesin hauls cargo toward the plus end of the microtubule, while dynein hauls cargo toward the minus end. Speeding vesicles recognize their specific target membranes through surface proteins, called Rab-GTPases, which initiate tethering upon arrival at the correct destination. Membrane fusion occurs when a V-snare protein on the vesicle locks into a complementary T-snare on the target membrane. To overcome natural repulsion, these snare complexes intertwine, generating massive thermodynamic force that pulls the lipid bilayers together until they merge. Once fusion is complete, the NSF-ATPase enzyme expends energy to physically pry the snare complex apart, resetting the system for the next vesicle. Intracellular movement and membrane fusion are highly active processes governed by precise protein-protein interactions. Operating this manufacturing network generates waste. Without an active clearance protocol, defective organelles and macromolecules would accumulate and halt production. The cell degrades this material through autophagy, utilizing the lysosome, a compartment filled with hydrolytic enzymes. To maintain these enzymes, VATPases continuously pump protons across the lysosomal membrane, keeping the internal environment at a corrosive pH of 5.0. Similarly, peroxisomes neutralize chemical hazards. They utilize the enzyme catalase to break down dangerous hydrogen peroxide produced during lipid metabolism into water and oxygen. Without these compartmentalized maintenance systems, the toxic byproducts of the cell's own metabolism would rapidly destroy its internal infrastructure. Beyond spatial architecture, this entire network is strictly governed along a temporal axis. This chart shows the cell cycle functioning as a genetically controlled phase space, synchronizing the duplication of every cellular component. The cycle begins in G1, the longest period of RNA and protein synthesis. Progression requires sufficient mitogens and energy, otherwise the cell halts and enters a quiescent state known as G0. If conditions are met, the cell enters the S phase, the singular window where DNA replication and histone synthesis occur. The G2 phase serves as the final preparatory stage, verifying that all energetic reserves and infrastructure are ready for division. Only then does the cell enter M phase, or mitosis, for the mechanical segregation of the duplicated components. Cellular division is the culmination of a strictly gated sequence designed to verify total system integrity before replication. Viewed as a whole, the system follows a clear pathway. mRNA exports from the nucleus, translates through the ER, modifies in the Golgi, and accelerates along microtubule highways. The ultimate mechanical test of this infrastructure is cytokinesis. The eukaryotic cell survives through the integration of these three pillars: compartmentalized chemistry, physical infrastructure, and temporal gating. At the nanoscale, cellular biology adheres to the principles of a strict engineering discipline. Decoding the logistical flow of information, matter, and energy at the cellular level provides the prerequisite for understanding higher-order human physics.